Today on ImmunoTea Time, we're joined by Professor Dr. Lars Nitschke, a professor at FAU Erlangen, Nuremberg. He leads a research group at the university working on B-cell functions and inhibitory receptors of signaling. As the immune system often runs without inhibition in autoimmune diseases like lupus, arthritis, and osteoporosis, understanding how these receptors work is crucial in finding new treatments and revealing the pathology of these diseases. Professor Nitschke has also received multiple awards for his wide-reaching work in immunology and has played a large role in elucidating how our bodies control the cells which produce antibodies. Lars, welcome to ImmunoTea Time.
Thank you very much. I'm happy to be here.
Cool. So today we have what was it called?
Iced wine tea, and Avanti gave it to us. So Avanti is one friend of us that is also studying with us, and so it's made with special grapes from Canada, and it's supposed to taste sweet. So we will try it during the
special winter tea
chapter, and we will see.
So Lars, you actually started out as a chemistry student, right? Not as a biology student?
Yes, that's right. I studied chemistry at the University of Freiburg and was first very interested in chemistry, anorganic chemistry, especially organic chemistry. And during the studies of chemistry, then I realized that I'm not so interested in synthesizing compounds for the rest of my life. I'm much more interested in biochemistries, which I then took as a major in my chemistry course. Mm-hmm. And then I had the chance to spend 1 year abroad at the University of Sussex in Britain, where I basically had freedom to do what I wanted. And during this year, I really took molecular biology courses, lectures in molecular biology and cell biology, and also worked in a molecular biology lab. And that helped me to make a change from chemistry to biology.
And then you moved from biology to immunology. And what sparked you to go through immunology?
Yeah, so first I did my diploma thesis. At that time it was not master's, but diploma. And the diploma thesis was in a structural biology lab, which crystallized protein to solve the structures. And I cloned a bacterial gene in order to determine the DNA sequence, and that was interesting. But then I realized if I would stay in this lab, I would be dealing with solving protein structures, which was somehow very functional and theoretical to me. And I was much more interested in biological processes, how cells work and how cells interact. And then I applied to different positions, both in Freiburg and in Heidelberg University. And by chance or with some luck, I was taken to start this PhD position at the Max Planck Institute for Immunobiology in Freiburg, headed by Georges Köhler, who had some years before got the Nobel Prize for the discovery of monoclonal antibodies. And that was a good chance to do a nice PhD thesis. And that's how I ended up in immunology. It was a bit coincidence involved.
Yeah, that must have been crazy at the time, having your supervisor get the Nobel Prize a couple of years before you start working with them. Right?
What was that like?
Yeah, it was, of course, a chance to work in this lab and a very nice opportunity for me, and also a bit unusual as a chemist to start into immunology. And to be honest, I didn't know very much when I started my PhD thesis. And then he also asked me to do a project about a new technique, which was just invented very recently before, the generation of knockout mice. And that was basically a jump into cold water because I didn't know much about immunology. And he said, this is this nice new technique, and I want you to do such a project. But I cannot really show you the technique because it's so new. I don't know it myself. But there's another PhD student, and you two together will find out how to do it.
Mm-hmm. I mean, it must have been. I mean, you're going really deep into immunology because you're working on IgD, right? That's like a.
Yes, so that was the project, to generate an IgD knockout mouse, because IgD is a very peculiar isotype of antibody which is expressed on the B-cell surface for while the B-cells mature and is always present, but it's not secreted. All the other antibody classes are also secreted as immunoglobulins, and IgD is not. So no one really knew what the function is. Why do B-cells express two B-cell receptors, IgM and IgD, and there must be a function. So with this new technique, there was, of course, a hope to understand the function of IgD. And so with a lot of effort, I generated this IgD knockout mouse, but unfortunately, it didn't have much of a phenotype. So these mice without IgD could make pretty normal B-cell development, B-cell maturation, and normal antibody immune responses. So it was a sort of disappointment at this stage that IgD is there and is conserved through evolution, but apparently not absolutely crucial.
Yeah,
that's the other. Yeah, it's the other part of science too.
Yeah, in my undergrad, we always had to learn all the antibody types like IgG, IgA,
IgM.
That's like one of the first things you get taught, like what all these antibody types are, and they always talk about IgD.
And then nobody says anything.
Right.
Yeah, it was only later on that certain functions of IgD were discovered, so it's really expressed in a distinct way on the surface to IgM. They are in different microclusters, probably have different functions because they can have different capacities how to bind the antigens. IgM is more versatile with monovalent or multivalent antigens, whereas IgD is more restricted to just multivalent antigens. So there could be a function in the type of immune response which is triggered by the two types of B-cell receptors, but the details are still not very clear.
So do you think during your PhD and your postdoc, or when you were starting out as a scientist, was there someone who sort of guided you as a mentor? Was it mostly your own motivation going into research, or what?
Yeah, because you also changed from chemistry to biology to immunology. So it was just yourself, or did someone guide you a bit?
Yeah, somehow immunology was quite interesting to me because it really covers a lot of biological processes, I would say. Communication between cells interacting with each other, cells migrating towards each other, being activated, one cell being activated by another sort of cell. So it's an interesting system to understand a lot about biological processes in general.
You think we should just follow what we're interested in? That's the advice for young scientists?
Yes, you should follow what you're interested in, and then some of your decisions are also a little bit driven by coincidence. This is also, you cannot plan everything. Yeah.
I guess
that's a nice advice.
I mean, it's just like
I overplan.
It's biology, like it's chance and what you need, right? Now that you're like a leader of a research group, do you miss being in the lab?
I sometimes miss being in the lab, yes, but it's very nice to supervise PhD students and postdocs and being close to their research projects and being able to discuss research projects. And so in a way, I feel I'm still pretty close to the daily research they are doing. And then I had the chance and took this chance to do sabbaticals during my time as a professor. And I did that 3 times, went to different countries, and in each case used the possibility to work in the lab, which hosted me. And really used the time also to do some practical experiments again, which normally I don't have time for.
The most recent one would have been, the year before last year, or was it last year?
Last year in Stockholm, Karolinska Institute, yeah.
So do you think, I mean, you've had the opportunity to do that, do you think science has changed a lot since you did your PhD, or things are similar? Like, how's the experience doing that? Like, I guess the process is the same, basically. You do an experiment and you collaborate with others, but, I mean, you've had like a long career in immunology, so some things must be different.
Yeah,
it depends a lot also on the group you're you were working in. So in this Max Planck Institute in Freiburg, people were thinking they are very good and they can do everything on their own and they don't need to collaborate. They're very independent, and so everything which occurs as a scientific problem or technical problem has to be solved within the group. And I think this is not how science is done these days. If you these days have a experimental result and you don't understand it, or you want to do a new technique which you're not familiar with, you start to collaborate with other groups, and you're much more open towards collaborations and exchange of techniques and getting supplementary techniques from others. And this is very much depending also on the scientific environment. So here in Erlangen, this is quite well working. Whereas in Freiburg, I was in an institute where it was not well working, and that was not good, because there are always limitations. Everyone is specialized in certain techniques and certain research questions, but cannot cover everything, and therefore it's a very good idea to collaborate.
Yeah, actually, in all the interviews that we have done, this collaborative thing of science, it has something that it has been mentioned, if not in all of them, in most of them. And I think it's something very cool that maybe you have a database and you don't know how to analyze it, and then you go to the bioinformatic lab and ask them, can you help? I think that's something very cool.
Yeah, and I think the German grant system, research grant system, fosters this a lot, supports this a lot, because what is special about German grant money organizations like the German Research Foundation is that they support very much collaborative grants. There are research units, there are collaborative research grants, and there are research training grants. All of these are grants given to several people who work in a certain field on a certain topic and are expected to work together on this topic. And this is a big advantage, I think, of the German funding system, that this is so much encouraged here.
Like the B-cell research consortium you were the head of, right?
Yes, I had the chance to also organize such a collaborative research consortium for 2 funding periods up to 9 years, and that was focused on B cells, regulation of B cells in both immune responses and also autoimmune diseases. And it covered 6 to 8 groups from Erlangen, 6 to 8 groups from Freiburg, and 6 to 8 groups from Berlin. So the German Rheumatism Research Center and Charité in Berlin. And that was a great opportunity to bring together all the main B-cell groups in Germany. And it was different fields: signal transduction, regulation of immune responses, also medical doctors working on rheumatic diseases, and a very nice mixture of different research questions, all focusing on B-cell responses and antibody responses.
And now to step back a bit and bring everyone on the same page, let's talk a bit about what is a B-cell. So we know that it just delivers antibodies, and we have had it in different chapters of the podcast. But now that we are with a specialist on B-cells, can you tell us what is a B-cell?
Yeah, so a B-cell is one of the 2 important players of the adaptive immune system, which can generate adaptive and very specific immune responses to pathogens, bacteria, viruses, and other pathogens, and can generate long-term protection against these diseases by antibodies, which are produced by differentiated B cells called plasma cells. And these plasma cells can remain in the body for a number of years. So these antibody responses can, depending on the virus or the bacteria, last for several years. Plus, they can also generate memory, which is then very important if you get a reinfection of the same pathogen, to get a quicker and faster and more efficient immune response than the first time.
So historically, when you got a vaccine, the B cells would be the important players, right?
Yes. So most vaccines are really working production of protective antibodies. So of course there's always T cell contribution to many vaccines, but most vaccines were designed in a way to produce efficient antibody response, IgG response, which are long-lasting and efficient.
And I guess when we talk about our bodies, or like the cells that stay with us for life, we talk about our skin cells, they have like a short turnover, or like our neurons, which I think most of them, from what I understand, they stay in us for our whole lives. And the plasma cells, they pretty much do that as well, right?
Yeah.
So it really is like a kind of memory. I mean, it's just like neurons. They stay there for a long time after you've encountered something. So, and the B cells are also important in disease, or like autoimmune diseases, right?
Yeah, so during my postdoc time in Freiburg, where I remained in the same institute, I was then starting my own project, really. That was my own idea to work on an inhibitory receptor called CD22, which was first characterized shortly before. And I was quite fascinated by a receptor which is not triggering immune responses, but rather controls it or inhibits it in a way to turn down the immune response when it's not needed anymore. And that's one of the complications which can happen if the immune response is not regulated in a normal way. There are different levels how to do that. And B cells seem to have these inhibitory receptors. CD22 is one of them, which inhibits the signaling cascade after it's not needed anymore, turns it down, terminates it, and therefore prevents an overstimulation. And overstimulation can, one consequence can be an overstimulation of the immune cells, which then get autoimmune phenotypes and start to attack the own body, for instance, by producing autoantibodies.
So normally you like encounter a virus or something, and then the B cells multiply, and then the virus or the bacteria is gone, and then is that when CD22 comes into play, or is it at the beginning when the B cells first encounter something?
Yeah, so in the case of CD22, it's rather the control of the tolerance versus autoimmunity. So prevents in the maturation of the B cells. That's my interpretation, that they're not, that autoreactive B cells are not overstimulated. There's always a small level of autoreactivity, even if there's tolerance mechanism, which during maturation of the B cells are thought to wipe out autoimmunity. Some weakly autoimmune B cells remain, and there's quite a few of them in the human that has been shown. And this is controlled by such counter receptors such as CD22.
So the CD22 is like a very big discovery in immunology, because it's just showing you that if we don't have this inhibition of the B cells, we will create autoantibodies.
Yes. So we generated CD22 knockout mice, and they have a tendency, and we have also characterized, and together with a group, I was collaborating with Paul Crocker's group, we were finding a second inhibitory receptor called Siglec-ting, and we did the first knockout mouse for Siglec-ting, and particularly this mouse had, when the mice were aging, an autoimmune phenotype with a lupus-like autoimmune disease. And this is even stronger if both of these inhibitory receptors are missing, CD22 and Siglec-ting.
And is there any role of CD22 in cancer? Because you have B-cell cancers, right?
Yes. So we also recently studied the role of more of Siglec-ting. We started with this one in leukemia, chronic lymphocytic leukemia. And the CLL disease, this is driven by also B-cell receptor signaling, and there was indications that this is crucial for the maintenance and the generation of these leukemic cells. We studied the role of Siglec-ting in a mouse model for this type of leukemia, a CLL mouse model, and saw in the Siglec-ting deficient mice that they have earlier developing leukemia and a stronger leukemia. And furthermore, we also generated mice where Siglec-G was overexpressed, and there we could suppress the leukemia to a certain extent, so it occurred much later. So showing that there is a role of these inhibitory receptors also in leukemia formation, because it could the lack of these receptors could lead to an uncontrolled proliferation, and then together with other acquired mutations, cancer may develop.
So the stimulation of the inhibitory receptors could be a treatment for leukemia.
Yes.
And the long run.
Yeah. So this is what we are currently addressing. So what we need to find is either agonistic ligands or agonistic antibodies which trigger this inhibitory function, and thereby maybe suppress this type of disease.
Would that be useful in autoimmune diseases as well?
Yes.
The same thing?
Yeah. So this overexpressing mouse, also Siglec-G overexpressing mouse, and we have a similar CD22 overexpressing mouse with just 5 times more of these inhibitory receptors on the B-cell surface. They also suppress lupus in a induced lupus model, pristine induced lupus. There also the induced lupus disease was lower than in control mice. Yeah.
And how do you find these agonists?
Yeah, so these 2 Siglec molecules, CD22 is also from the same family. It's called Siglec-2 as a second name. They bind to certain types of sugars, sialic acids, and this is very low affinity binding, and these sialic acids are also quite abundantly expressed on many cells. But together with chemists, we have found high affinity ligands, which are sugar analogues, sialic acid analogues. And we are testing these sialic acid analogues, which have high affinity binding, whether they are agonistic. And we can do that by simple readouts, such as calcium signaling. If it's agonistic, it would suppress calcium signaling. There was already an agonistic antibody against CD22 called ebratuximab, and that was tested as a possible new treatment for SLE, for lupus disease, but failed in phase 3 clinical trial. Yeah, so there was also hope with this first antibody that it could work, but yeah. But maybe antibodies for Siglec-G or the human counterpart called Siglec-10 are more effective, or agonistic ligands.
Yeah. And you also worked with Miriam with the toxin conjugated antibody. So what was that about?
Yeah, so we coupled CD22 ligands, also these sialic acid analogues, to exotoxin A, which is a toxin, bacterial toxin, in order to use CD22-positive lymphoma, B-cell lymphoma, or leukemic B-cell leukemic cells to target CD22 and introduce this toxin intracellularly and thereby kill leukemic cells and lymphoma cells. Yes.
And did it work?
Well, this was not entirely successful. We didn't see much effect of these toxins, but this could have just technical reasons. We started first with certain mouse models, and there we didn't see much of an effect. Yeah,
That's quite interesting.
Yeah. I mean, it's like a different sort of antibody therapy for cancer, right?
Yeah.
Not just like a signaling.
I mean, at the end now,
antagonist or something.
with everything related to engineering in biology, you can couple everything, and I think you can achieve a lot of things.
Yeah. Of course, the checkpoint inhibitor antibodies for the T cells, they are basically a similar approach, and the B cell side has not been covered so much. So, in a way, it could be a possibility to target B cells in this way.
Why do you think B cells haven't had as much attention in that regard, like the checkpoint inhibitors or the CAR T cells? Or I mean, it's always T cells, right? I guess because of the what do you think?
Yeah, but think about the targets for the CAR T cells.
I was going to tell you. CAR T cells target B cells.
I mean, this is a real surprise. I mean, these Erlangen studies with the CAR T cell therapies, I mean, they are, of course, addressing a B cell protein, CD19, as you know. And it's quite surprising also to a B cell immunologist how effective they are, because a lot of people, of course, we are quite convinced that B cells play a very important part in diseases like lupus, where anti-DNA autoantibodies are so important. But other people questioned this and said, lupus is a very complex disease and has many players and many immune cells are involved. So it's quite surprising that you just kill the B cells or the plasmablast with this therapy and get such an effect, such a drastic effect, and also in other related diseases. And also in diseases like multiple sclerosis, where in MS people would have thought macrophages and T cells are the immune cells which attack the neurons in the brain, but surprisingly there the anti-CD20 antibody was quite efficient. So also there, it's also another example of autoimmune disease where B cells seem to be quite crucial. Yeah.
Would antibodies targeting the signaling of B cells, it's sort of less extreme than a CAR T cell killing the B cells, right? So would it have less of an effect in a treatment for something like lupus, where
it's difficult to predict, but it's of course potentially transient, and it would not kill the B cells, it would just modify the signaling. But if that's enough, you would certainly have to give agonistic ligands or antibodies continuously. But it would just be an alternative, I think, to killing of the B cells, which has, of course, long-term side effects potentially, because then you lose the B cells completely and may have problems with infections and so on.
Yeah, also with the memory, they are like, it's not very well known yet. So just maybe for like leukemia or some cancers, maybe CAR T cells are better, but the antibodies maybe for autoimmune diseases that are not that life-threatening, maybe in the end is a better option. We don't know. We will see in the future. What do you think? Because like in the past, we have an understanding of B cells in a way, and maybe now it changed. Do you think it changed? How do we understand B cells, the importance that we give to B cells?
Yeah. So I think there has been a lot of development in the last years, because before it was thought that B cells are mainly producers of plasma cells and antibodies. That's their only function. But now it's been pretty clear that B cells have other functions than just antibody-producing cells. For instance, this involvement of B cells in MS and other autoimmune diseases can only be understood because it's not the plasma cells and the antibodies which are targeted here, but the B cells themselves. So they are either crucially involved in antigen presentation to T cells in the brain or in cytokine production. And that's another field that certain B cell fractions or plasmablasts can produce cytokines like interleukin-10, which are regulatory cytokines, and that's also a modulating function. There may be other plasmablasts which produce inflammatory cytokines. So there's more and more diverse functions, I would say, of the B cells, because they are also regulating.
So even taking away the antibody release of the B cells, they have other functions.
Yes.
Really, that's really interesting. I'd never heard of that before. The other functions of B cells.
Yeah. I mean, if you think about it, antigen presentation, of course,
you know, which is
triggering an immune response, a T cell dependent immune response. But this could be, just the only function they have during an autoimmune disease like MS in the brain, then it's not the antibody. There are also, of course, typical antibodies found, but rituximab and other antibodies which address CD20 work quite efficiently, which means, and they're not targeting plasma cells because the plasma cells don't have CD20 anymore. So therefore it must be one of these antibody-independent functions of B cells. I mean, it's not completely clear what it is, but the guesses are either antigen presentation or cytokine production.
So I guess even in that regard, using these like inhibitory receptors of B cells, it's not just the antibody functions you're altering, it's also everything else they do.
Yeah.
Right, which...
Yeah.
That would be beneficial.
Yeah. Or not beneficial, right? I mean, it's...
Depending on the context, yes.
Yeah.
I'm always thinking in the positive things. Yeah. Yeah, we also wanted to talk about in the chapter about the mentorship, because you are, like, for the people that don't know, you are a very important part of our master program. And how is leading a master program and being involved in it, like change from research in the lab to something completely, maybe not different because you are still bound to science, but...
Yeah, so this integrated immunology master's program is a special master's program. It's first of all international, which we appreciate very much, whereas all other teaching is in German. This is in English. And it's a very selected group of students. And we have this very nice funding by the elite program of Bavaria. And therefore we got a lot of positions and can do different sorts of teaching, and also mentoring is possible in a way like it's normally difficult to do with 200 bachelor biology students. 25 integrated immunology master students are with so many professors and lecturers are more easy, or it's more efficient and possible in another way to supervise and educate them, I would say.
Yeah, I think sometimes we can be a bit difficult as well.
Yeah. Not really.
But it must be different to working with a PhD student or like a postdoc, right? Because that's also like scientific mentoring and education, doing that, right?
Yeah, to be honest, it's difficult for us if we do teaching to large groups of students and we don't get to know them very well, because then we are standing in these big lecture halls and giving our lecture and ask questions, and no one dares to give an answer. And no one dares to ask a question, so there's not much contact. So it's much more familiar in a course like this, and this we appreciate, I mean, it gets better in the usual courses in the higher semesters, because then we do the practicals, and in the practicals there's more contact. But if there's not a good contact to the students, that's also frustrating for us, because we don't know if they listen to our lectures, if they're interested, if they understand it, if they're totally bored. So all this type of feedback is important, because it's much more rewarding and much nicer to get feedback than to do teaching in an empty space. That was particularly bad during COVID time when we gave these online lectures and had all these Zoom meetings where the cameras weren't turned on, and it was like talking into the dark space, and then the time was over and there was nothing, not even eyes to look at. Very strange.
I have a question actually for the COVID time, because here in Germany, when you finish the lecture, you knock at the table, right? During the COVID, they also knock? No, they didn't.
There was nothing.
Oh, that's sad. Because I remember the first day that we came and had the introduction, and then all the Germans started knocking the table and we started clapping. It was a bit confusing. Then we realized that we have to knock the table. That was fun. But talking about the not having this feedback and stuff, we are very lucky, because in our program we have this problem-oriented learning courses. And I think you introduced them into the program.
Yeah.
Like maybe you can explain, because when you read the program of the master, you find polls and what is this?
Yeah. So you all didn't know this. Even you didn't know this, Ben, from Australia, this type of teaching?
Not really.
Not really, okay. Yeah, so we got these positions for this elite program, and they expected us to do more advanced or modern, unusual ways of teaching as well. And by chance, my son studied at the University of Maastricht in the Netherlands, something totally different, economics. And Maastricht University is known for this problem-based learning, how they call it. It's the same, the English expression, problem-based learning or problem-oriented learning. And he liked this very much, the small group teaching and these tutorial groups, and told me a lot about it. And I thought, that would be really nice to try to do that in biology as well. But it was somehow very difficult with thinking of groups of 150 students, and you need all these tutors for these little groups. And when this program started, I got the idea we could introduce it here as well, and it took a while to persuade my colleagues here that we should really do that, especially the colleagues from the medical faculty. They were quite skeptical, this is not a good way of teaching. And then by chance, Klaus Überler had been at Bochum University before, and he knew the study dean there, and he knew that they have done this problem-oriented learning in medicine in Bochum. And the study dean came first for a talk, and that was already quite nice. And then he did a workshop with us, with all the professors from biology and medicine, where he gave us a case, like the first poll session, like you know it, and then all the professors had to do a brainstorming and solve the case and develop some learning goals, and they all had fun, yeah. And then after this 3 or 4 hours with introduction and the workshop, then everyone was convinced, and then we just decided to try it. And since then, we've introduced it, and it's working quite well, I think. And the students like it, and all the tutors who supervise these groups usually like it as well. So I think, in my eyes, it's a very nice addition to the lectures. Maybe not the only way of teaching, but a very nice addition where you can get deeper in certain fields. And the self-directed learning of the students works very well. In my experience, it's nice, especially because of 2 aspects. The students coming here come from very different backgrounds. So they have had immunology before or not at all. They've studied biology or biotechnology or totally different subjects. And they come from different countries with very different teaching traditions. And so therefore these tutorials, these groups help them to integrate their knowledge, which they have brought with them, particularly during this brainstorming part, because they all can write down their ideas to the case, and it's very often very good ideas, even if they are quite quiet in groups. And then they realize, they can contribute themselves as well. And then everyone gets a discussion leader with change roles, and everyone has to do it. And in this way, the group atmosphere of these tutorial groups is quite good. And the groups grow together. That is my impression as a tutor. And it usually helps the international students to integrate quite well.
Yeah, that's actually the case. I mean, both of us, we are not from here. And when we arrived to a very big class, but not very big because we are only 23 in this year, but we are a big group of people, and then you kind of maybe don't know with who to talk or like how to integrate well, and then you find yourself in a group of 5 people and everyone has to talk. Yeah. So at the end, I think, maybe not that much for me because I'm a very extroverted person, but maybe for other people that helps a lot and brings the group together. And at the end, like the class is kind of very like a pineapple. We are all together.
And also the more quiet people, they gain confidence from week to week. And this is also nice to watch as a tutor. Usually we don't do very much. We just watch the group and don't interfere so much. But it's nice to observe how they get more and more confident and then contribute to the discussions. And so I think it's a very, very nice system.
Yeah, I mean, it's fun with the problem solving. Like someone comes in and they have some fever or something and a bunch of symptoms, and then you can try and figure it out. Or I think the best part for me was we come in as a week of like 5 or 6 students, and then we get this prompt. Yeah, like someone has dengue or some disease, and then we all tell each other what we know already, and then we can figure out what we want to know all together, and then we come back the next week and figure it out. But I think a lot of the times, if you're doing something like that, like a task in a group, there's a lot that we all know, or that most of us know, and then, I mean, we all know this, but then we just talk about it anyway the next week, like instead of establishing what we know already, right? And then we just end up with like 20 minutes of a presentation. I mean, it's all stuff we know, but and then we're just getting repeated and no one's enjoying it. But if we all figure out what we want to know and what we know already, and we can come back the next week, and then it's actually interesting because you're learning stuff
It got better through time. It got better through time.
Yeah.
So first semester, we didn't know what it was. I have never heard this in my life. And then I found there myself finding about glycosylation, playing with papers. And I really liked it. I learned a lot. Yeah. And then second semester, we kind of knew what it was, how like we had new groups. So we kind of established what we want. So we just find like, okay, now we know what dengue is. Okay, then I want to know more about this and what about that. And then it was kind of like an improvement from first semester to second semester in the way that we saw how the problem was.
You can also set your own aims, how you like it as a group. And it can develop in different directions. It's not set that we expect a certain type of answers and problem solving, but it can go in this direction or in the other direction. So this is, it's quite flexible in a way. Yeah.
And you can do a lot of games. So for example, in my group in the first semester, at the last ball, we end up playing beer pong. So it was very nice. It was very nice. So it's just kind of way of learning and having fun and meeting professors. Because, I mean, at the end, it's always good to have connections, right?
Yeah, so I think the teaching matters a lot in a master's program. It's not just the lectures or the having to do a lab report or an assessment during the semester. It's good to have something that's so involved, I guess. It's good to have.
So now that we reached the bottom of our cups, it's time for the last tip question. This is 3 quick questions that we ask all of the guests. And the first one is, if you could go back and speak to yourself in the first day in the lab, what would you say to yourself?
Just stay curious, stay interested, and don't lose the confidence in the process.
That's very important. Like in the 4th semester, we are going to start a master thesis, and I think it's very important to don't lose hope and the curiosity.
Don't lose hope.
Yeah, it's important. I think it's important.
Yeah.
And then the second one is, what's one mystery, it can be scientific or personal, that you hope to see solved in your lifetime?
Yeah, so how to prevent the occurrence of autoimmune diseases? How can you develop therapies to heal these type of diseases very early when they develop at a very early point? And a question related to this is also, what exactly is the cause why they are so much increasing in the Western world? I mean, this is an observation, and they're much higher in incidence than in less developed countries. And there are theories to explain this, but it's still pretty much unknown, I think, what the real reason for that is.
And then the last question is, if you could ask the immune system one question and get a clear, honest answer, what will you ask?
If I could ask the immune system, why do you not establish a complete tolerance for B cells and T cells to prevent all types of autoimmunity? So is there any reason why you don't do that? So is this low level of autoimmunity maybe an advantage towards fighting certain infectious diseases? There's relevance. I mean, there's evidence that this cross-reactivity also has a certain function to broaden specificities. But this is a question I would really have. Why is tolerance induction not complete? Yeah? And why do autoreactive B and T cells remain in the immune system?
Mm-hmm. So if there was like a genie and he came up to you and he was like, Lars, I'll make a proposition for you. I'll make it so that you can never have any autoreactive B cells again. What do you think? Would you accept that? Do you think that would be good for you?
It's difficult to tell. So there must be a reason. I mean, in the B cells which come out of the bone marrow in the human, 20% or so are weakly autoreactive or polyreactive. So they have a lot of specificities against self-antigens. So there must be a reason. So I'm not entirely sure whether I would say yes to this suggestion. Maybe it's dangerous to be that strict.
Yeah.
Mm-hmm. Yeah.
Okay.
Yeah, and then one question, because you are a B-cell person and there is people that is T-cell person. What would you say a T-cell person to convince them that B-cell are better?
Yeah, so at my last position in Würzburg, I was one group working on B-cells in a total T-cell environment. Mm-hmm. And the boss of the institute was, of course, also working on T-cells. And he said, you know what B stands for in B cells? B stands for boring. Because the T cells are the real interesting ones, and the B cells just copy the function of T cells. And I would answer that, so there is all this evidence how important they are in vaccinations, and even what important role they play in certain diseases like autoimmune diseases, which is unexpected, that I would say they are at least as important as T cells, but it's always collaborations. Yeah? Mm-hmm. So these two types of cells have to work together.
Yeah. Mm-hmm. That was very diplomatic. I think the T cell people would be happy with that.
Yeah. Well, so thank you very much for the conversation. It was very nice. We learned a lot about B cells and about mentoring, that we know that you're a very good mentor. So thank you very much. I mean, you know it more. He's your mentor, so. But thank you very much.
Yeah, thank you very much as well. I enjoyed the interview.
Yeah. Cool. Until next time.
Stay curious.
Stay critical.
And see you next time.
Reach us on Instagram at ImmuneFAU or send us questions, ideas, or feedback to immunefau@gmail.com. The equipment for this podcast is graciously provided by the Friedrich Alexander University Rundfunkstudio and Thomas Bauernschmidt.